All right. Welcome and happy Tuesday. I am Kathleen Christ. I'm the Executive Director of Houston Area Parkinson's Society, but today I'm a representing member of Independent Parkinson's Network. IPN, as we are known, is a group of 17 regional nonprofit organizations from across the country who provide support and resources to our local communities and that work together to share best practices, valuable resources, and take collective action toward improving the quality of life for those impacted by Parkinson's disease. This is guided by a shared understanding that strategic collaboration will improve local organizational effectiveness and deepen national impact. A big part of that impact is through education and finding opportunities to bring the latest and greatest Parkinson's information to our communities. There is a lot happening in the Parkinson's space, and our goal is to make sure that you have the information necessary to successfully manage Parkinson's long term. And today, we are thrilled to welcome our partner, Ask Bio, to present an update on regenerative therapies for Parkinson's. Today, I think we are all very eager to learn more about the progress and promise of gene therapy, what it is, how it works, and even how we can get involved in moving that science forward. So, the way that this works is that our presenter is going to present. During the presentation, you are welcome to put your questions in through the Q&A function that is most often located at the bottom of your screen. And after the presentation has wrapped up, we will go through the questions that are relevant to the topic that we are discussing today. And there are hundreds of people on today's webinar, and so we may not get through all the questions, but we are going to do our best to get through the majority of them. So with that, I would like to introduce Pooja Merchant with Ask Bio and give her the floor to make a couple comments, and then I will introduce our speaker.
Thanks, Kathleen, and hello, everyone. Great to see such a high level of participation. I am Pooja. I work in the medical team at Ask Bio, and just want to say it's an honor for us to support this webinar. We are a gene therapy company, and we're always looking for ways to bring in information to the patient community in a way that you understand it, but also to partner with you and to co-create the research so that we're really focusing on the needs that matter to you, so that we can truly improve the outcomes that make the most difference. So thank you. Feel free to ask questions where our desire is to really make sure you leave with a good understanding of where the regenerative therapy space is going and the science behind gene therapy. With that, I'll hand over to you, Kathleen, to introduce our speaker.
Okay, wonderful. Thank you. All right. Well, your speaker today is Dr. Amber Van Laar. She's a movement disorders trained neurologist, but it's the potential of gene therapy that has really driven Dr. Van Laar's clinical and research interests in the development of novel gene therapies to treat previously untreatable diseases. Her early research investigated the disease-modifying potential of gene therapy for common and rare neurodegenerative conditions. These experiences led her to further explore gene therapy as a platform to treat both rare and common diseases, moving gene therapy beyond genetically based diseases. Dr. Van Laar continues her work with gene therapy and now serves as the Senior Vice President of Global Clinical Development for Ask Bio in conjunction with Bayer Pharmaceuticals. In this role, she continues to develop gene therapies that hold potential to alter the course of diseases with high unmet needs in the hopes of bettering the lives of patients and their families. She is a very good friend to the Parkinson's community, and we are so glad to have you with us today and welcome you.
Thank you so much. Let me get my screen shared here. Let's see. And I think I was hearing that there were some concerns with reverberation, so I just want to make sure everyone can hear me. All right. Get my marker up here. Okay, it's a real pleasure to be able to speak to you all today. I want to give an update on big regenerative therapies like cell and gene therapy today, and really give you a sense of what's actually in the clinic right now and what's on the horizon to come. So we're really going to focus in on cell and gene therapies. This is part of what we call advanced therapies for Parkinson's disease. And I want to explain, like, why exactly do we always lump cell and gene therapies together? And so typically these are therapies that are given in very unique ways. So these are often delivered surgically. We have to get around the immune privileged status of the brain, and that often means that we have to directly inject or administer the cell and gene therapies to the brain itself. This is also intended to overcome some of the limitations we have with standard of care. So meaning that we have these therapies that are really meant to get around and go above and beyond what we're doing with just standard of care things like levodopa, for example. The other sort of unique thing about what we're doing in cell and gene therapy is we're really looking for these types of therapies to reverse or slow down the disease progression. This means that we're looking for the potential for disease modification with these types of therapies and not only just treating symptoms. So if we're really going to be, investing in something like a neurosurgical approach for these therapies, we're looking for a big effect. And again, this potential to alter the disease course. So we're going to start with cell therapies first. We're going to dig in here first. I know there's a lot of interest in this, especially with a lot of the news recently. So let's just start with some basics. So what are we replacing and where are we replacing it? So we know that for many years now that in Parkinson's disease, the hallmark feature here is the loss of dopamine neurons in the substantia nigra. So the substantia nigra sits down here in the brainstem, and it's from the cell bodies here that then project to the putamen. And it's the loss of these cells, and they can't project the dopamine here in the putamen. And without dopamine, that's what causes the symptoms of slowness and stiffness and the tremor that we all see with Parkinson's. And the putamen here is really where the business end of dopamine occurs. It happens to be that the putamen is where we actually do a lot of the therapies we're going to be talking about today. It's a larger target. It's a lot easier to get to from a surgical standpoint. And so many places we're going to be talking about are here in the putamen. So that's where. Okay. So why would you want to do cell therapy for Parkinson's disease? Well, like I just said, in Parkinson's, this is a loss of dopamine cells. And we know in PD, there's a specific vulnerability of dopamine cells in particular. So again, these dopamine cells sit in the substantia nigra, which is here in the upper part of the brainstem called the midbrain. So if you took a slice of that here, this is what it would look like. And we're looking at the substantia nigra here. So it's actually just this purple layer here where all those dopamine cells sit. And it's really the loss of these cells that then leads to the symptoms of dopamine or of Parkinson's because there's a loss of dopamine. And so the idea with the cell therapy is we're replacing the cells that can't make dopamine and use them to make them work like they're little mini dopamine pumps. And that's how you can relieve the symptoms of Parkinson's with a cell therapy. So what are stem cells? I think we are probably all sitting here with an idea of what we think stem cells are, but in fact, they're quite varied. But stem cells, the best way to think about this is that they're raw materials. This is where all of the cells from the rest of the body come from. So one stem cell can turn into 200 different types of cells to help make a human, let's say. And this is how we have, blood cells, fat cells, how we get our muscles and bone, also our hair. So these cells can make just about any part of our body. Now, a lot of what we're doing from a cell therapy standpoint for Parkinson's is we can take some of these stem cells and actually force them into a dopamine producing cell in order to be able to treat with Parkinson's. So basically in a lab, in a dish, we can take something like a skin cell and then push it chemically into a dopamine cell. And that's in some cases what we're using for some of these studies I'm about to show you. So where do we get the cells? And this is, can be controversial in some respects, but there's lots of places that we can get cells that have been used in Parkinson's studies in the past, actually. So bear in mind, Parkinson's has been over a couple decades now has been looking at cell replacement. And in some cases, we used things like autografts. So these are actually cells like dopamine containing cells in other parts of the body. So adrenal gland, for example, had been used and see if that would help treat Parkinson's symptoms. There's also things like allografts. So this is coming from other humans. So in which case there were fetal dopamine cells in the past that have been used, and even some retina cells that can make dopamine have been transplanted in the past for Parkinson's. And even though it may seem very sci-fi, we have xenografts, so meaning that we have cells taken from other animals that have been transplanted into humans. And sure enough, in the past, we had pig dopamine cells that have been transplanted with some mixed results. Now, a lot of what's very popular right now, which is hopefully you've heard a little bit about these iPSCs. So these are cells that are derived from yourself, from an adult, and this in some cases is from skin. And that can then, again, in a dish, be transformed into a dopamine producing neuron. And then there's embryonic, which I think most people are familiar with, and sometimes are kind of synonymous with stem cells. But I hope you can appreciate there are lots of places we can get stem cells. It's not just embryos. And we'll talk about that a little bit more in the studies to come. So there are still a lot of challenges for cell therapies. As I mentioned, there's actually been cell therapy for Parkinson's being looked at in clinical studies for decades at this point. So why haven't we gotten further? What have the challenges been? And some of the big issues are really with manufacturing and how do you scale up? And eventually we get something that's approved. How do you get it available for the millions of Parkinson's patients worldwide? So this is still a big holdup for everybody. We also just don't know what's the best cell type. I just listed a whole bunch of different places we could make dopamine cells, but we don't know which one's the best one. Maybe there's a couple. And then where's the best place to deliver? How are we delivering it? These are still limitations that we're trying to figure out now. I mentioned there's some ethical concerns, especially with the embryonic stem cells, that can be a bit of a headwind. There's still immune suppression. So these are like transplants, like we still are suppressing the immune system when we're delivering the cell therapy. In the past, there was an issue with runaway dyskinesia. So this is dyskinesias or these involuntary kind of wiggly movements that in this case, they didn't stop. They were very difficult to control. And so there was a concern that this could happen again, though we haven't seen that in any of the recent studies to date. But beyond that, one other major challenge for all the companies developing these cell therapies is at some point they're going to have to prove to the FDA and payers and other groups that the cells are alive, they're working, they've incorporated, and they're doing what they said that they would do. And that's actually more challenging than it would seem. So there's still a lot of questions that remain right now for cell therapy. So what's the right dose and what's the right type of cell? This is still a big unknown, and I think you'll see here in a minute that there's a big variety of what's being looked at right now. One other issue, it's the brain. This isn't like a bone where you can just repair it. So how do you get the transplanted cells to get into the native network and start connecting and making all the appropriate connections and discussions between cells? That's actually very hard to know and very hard to we can't image it necessarily. It's a little bit difficult. Is there a right type of PD patient that should go on to get cell therapy? Is there a better way to deliver? How do we get this reproducible? How do we make it last? And I think another big question in the field right now is, if we have to do we have to suppress the immune system? And if so, how much, how long, what drugs are we using? All these things are still very much being researched right now in these clinical studies. Okay. Going to move on now, and I just want to go through. There's only a couple of cell therapy studies that are active right now. I'm primarily going to be showing you studies that are available in the US and that are active right now for cell therapy, specifically for Parkinson's disease. So this first one we're going to be talking about is looking at a cell therapy from Blue Rock. This study is active. They're still collecting data, but they're no longer recruiting for the study. So this is a phase 1. It's open label and dose escalation, meaning that everyone knows that they were being treated with active cell therapy and that there were 2 doses that were being looked at. As with many of the studies I'm going to show you, this is neurosurgically delivered to the putamen, which we just saw a little bit ago. And in this particular study, they were using embryonic-derived stem cells. This data has been published, and there were some great results. And in the high dose group with using 2.7 million cells, when they look at this group at 18 months post-transplantation, there was some pretty significant motor improvements. So notably, some increase in good on time, less off time that corresponded, but also looking at the UPDRS. So this is the neurologist measure of how severe the Parkinson's symptoms are. In this case, it improved by over 20 points, which is very significant. A high level of change in the UPDRS is about 10 points, and this is double that. So this was very significant. And in fact, in summary, this looked, the procedure and the cell transplant looked to be safe and well tolerated. So was the immune suppression that was used at this 18-month time point. And I would say the company was very, excited by this data. And, though it's a small open label study, it was enough to go on to make this study. So this is the phase 3 study from Blue Rock that's currently open. It's active. It's recruiting. They're looking for participants right now. So some similarities to what I just showed you. So it's a controlled surgery this time. So some patients will not get active cells being transplanted, and it's double blinded. But again, the similarities, we're replacing dopamine neurons in the putamen using the neurosurgical delivery, and we're using adult dopamine neurons in this particular study. You can see who they're looking for and where they're doing the study. And some upsides in the following slides, we're going to have upsides and watchouts. And the upsides here are you're replacing directly the dopamine cells that are missing in Parkinson's. And therefore, it really should be helpful in relieving the dopamine responsive symptoms, which we just saw from their results. But with all cell therapies, we have to, be careful and make sure that the cells survive and that they're working and they're incorporating with the rest of the brain network, but also looking at the immunosuppression aspects and how that's working long term. So a really exciting story, and phase 3 is going on. So keeping our eyes on how this study goes. Another study that's out there right now, again, active and enrolling right now, it's only enrolling by invitation. So this is looking at autologous cells. So again, coming from self. And in this case, they're using the patient's own skin cells that are transformed into dopamine producing cells. So this is a phase 1, so very early, it's open label. Everyone knows that they're getting active treatment. They're again doing the neurosurgical delivery to the putamen, just like everything else we had seen. You can see the adults they're looking for and get a sense of how severe the Parkinson's must be. And this is a US only study with Aspen Therapeutics. So again, with cell therapies, you're again directly replacing the cells that are dying because of PD. And again, we need to watch very carefully how well these cells survive and do they graft well over time. But one issue here is the scalability. It's very difficult to take, a biopsy from a patient, convert all the cells, to the dopamine cells, and then replace them back, a few months later. So it's something to watch. Very fascinating. This is clearly as precision medicine as you can get. I guess one other marker here is I don't think that they're going to be using immunosuppression in this one because it's your own cells. There's just one last cell therapy that's out there right now. So this is, again, active recruiting. This is probably one of the newer ones right now. Again, a phase 1/2, very early, open label, meaning everyone knows what they're getting. And it's just a one-time delivery. It's, again, neurosurgical delivery to the putamen. And you can see the type of cells that they're using here coming from the patient's own skin cells. You can see, again, very similar to the patient profile we just saw with the ages 50, 70, and more than 4 years. And this is only in the US. One interesting thing about this study, these are cells from Sumitomo. This was actually approved in Japan very recently and based off of a very small patient study. So they're coming here to the US, doing a full study here in the United States population to see if those results hold up here in the United States. So interesting, very promising for the field of cell therapy. And again, something we're watching very closely. And I think it's just nice that, again, for all the history we've had of cell therapies in Parkinson's disease, I think we've really made some major headway and really learned a lot of lessons that over the past couple decades. Just one little watch out I want to, I always like to share this piece. There's a lot of concerns for medical tourism. So there are a lot of cell therapies for Parkinson's that are out there in the world. And just as a warning, just a watch out that not all studies are done, to the same rigor and the same stringent standards that we have in a lot of other studies that I've just showed you. I always encourage patients to make sure they look up any clinical study that they're looking for. They must be listed on clinicaltrials.gov to be a legitimate study. So just be careful, with what you're looking at and where you're getting your information from. So, and talk to your doctor. Okay, I'm going to move on now to gene therapy. So again, very similar, different mechanism of action, but this is near and dear to my heart. So it's just like we did with the cell therapies, we got to have this kind of start and level set. And so I like to always start here with what are genes? Just make sure we're on the same page here. So genes are pieces of DNA that can carry the genetic information from generation to generation. And the Human Genome Project that wrapped up many years ago now, we identified about 20 to 25,000 genes that we each carry. But of all those tens of thousands of genes, only 1% of them is what really makes us look so different from human to human. And it's really interesting that such small changes makes us look so different. And I think what's really important to kind of keep in mind is that every cell has DNA, and every DNA has some genes in it. And with each little piece of gene, that then gets read by the cells and turned into a protein. And it's really the protein that does the work, let's say. And so just keep that in mind as we get through the next couple of slides. So doing a little bit of schoolwork here, going to do some gene therapy 101. So it's helpful to think of genes like instructions or a blueprint. And these are instructions then for all of the health and growth that's needed for every cell in your body. Now, every cell, this is just their normal machinery. They read the genes, they read the instructions, and then they make a new protein. So what is a protein? So proteins are substances that are used by the cell to signal the health and growth and many other functions in the cell to help keep the cells alive. The proteins are also things that give your eye, the different colors that we see, and your skin, the different, structures and color. And it's really with gene therapy, what we do is we take advantage and we harness that normal machinery. The cells normally read genes and make proteins, and we're using that, harnessing that with gene therapy to then make a protein that we want, and using that to then act as our medicine. So telling you this in a slightly different way. So how do we deliver genes to different cells? So with gene therapy, we use a healthy copy of DNA, so whatever gene that we want to put in. And that means that we are not limited to just genes with known mutations or diseases with known mutations. We really could treat something like Parkinson's that has lots of genes involved, for example. And in this case, in most gene therapies that are out there today, they use something called AAV, or adeno-associated virus. This is a virus that's been modified and can't make people sick, but it really works well as a shell to help get our gene in. And so what we do is we have to package our gene of interest inside this AAV, this capsid here in red. And it helps to think like our gene therapy, like the gene of interest is our letter, and that the capsid is like an envelope. And we have to package these all together and then deliver that to the brain. And it happens to be that AAV is really good at delivering these instructions to brain cells specifically. They're really hard to get genes into. So breaking this down a little bit more, so how does this work? So again, using this analogy, think of our gene like a letter. It's a set of instructions that we want to send to the cell. Just like we want to send a letter, we have to package it up, and we get our full package, and then that needs to get delivered to whatever part of the body we'd like to deliver this. Sometimes it's IV. In our case, we're talking about delivering it right to the brain. Now, we know that this shell, this envelope, is really good at getting into the cell that we want. That's what AAV is really good at. And so this goes into the gene is then put into the cell. And our envelope, just like you would with another letter, the envelope gets thrown away, and all that's left behind now is our little piece of gene. And from that, the cell does what it always does. It reads the instructions and makes a protein. And this is the basics of how gene therapy works. Breaking it down again. So we got our gene, we need to package it. Once it's fully packaged, for CNS, or for the brain diseases, we put it directly to the brain. And again, all we're left with is a little piece of DNA that we put in there that we wanted. That's a healthy copy. And then we get the protein made from what we want. And it's really this protein that then does the work inside the cell itself. Okay, so I'm going to dive into some of the gene therapies that are going on right now for Parkinson's disease. So this is AAV-GAD. So this is also active, but not recruiting. So they've recruited the full study and are just collecting data at this point. So it was a phase 1/2, and this was a double-blinded study. So again, some patients did not receive active treatment. And looking at overall, just this is safe, and can we do this? Now, this is a very interesting mechanism of action. So really trying to change and alter the circuitry. So the way that the different parts of the brain are signaling to each other as a way of combating the Parkinson's symptoms. So very different than what we've seen before. And in some cases, they talk about it like a DBS, like gene therapy as like a DBS. So because of this, they're going to, again, it's neurosurgically delivered, but we're going to a different part of the brain here called the subthalamic nucleus, which actually is a target fairly often for DBS, for example. Again, the hope here is that you normalize the electricity that's happening between these different brain regions in order to make it look more normal and more like you don't have Parkinson's. You can see the types of Parkinson's patients they're looking for here, and this is a US only study. It's really, again, this is the very unique mechanism of action. So this trying to, again, change the circuitry, the signaling between areas. There's really nothing else like there out there right now in gene therapy. Just keeping an eye, we're watching out, want to see the results, and it's still really not clear what the crossover it looks like here. So again, really waiting for some of the data to come out from this very promising study. So there's not too many more, but this is another interesting and kind of unique gene therapy approach for Parkinson's disease. So this is AAV-GBA, and there we're looking at patients with a known GBA mutation. So this was very limited to a very small subset of Parkinson's patients known to have a GBA mutation. So again, a very early, so phase 1, 2, open label, meaning everyone knew what they were getting, and they did higher and higher doses in this particular study. So just a little bit on GBA. So this is part of the garbage disposal system in Parkinson's. The idea is that if you don't have enough GBA or if it's mutated in some of these patients, it doesn't work as well. And so all the junk and the proteins that get messed up, they're not able to be sequestered or destroyed appropriately. And this is where things like Lewy bodies might come about because you're not able to process those damaged things in the cell as well. So the idea is that when you do GBA gene therapy, you increase healthy levels of GBA inside the brain. And that should then hopefully combat some of the issues by not having GBA working in the first place. What's different about this one, so they use a cisterna magna infusion. So they get to the spinal fluid at the base of the brain. So there's actually nothing going into the brain itself. So that makes it very different. But it's still a one-time delivery and it does require some immunosuppression still. You can see the types of PD they're looking for. And again, you have to have had a GBA mutation for this particular study, which I think is going to be very interesting to see when this reads out. And you can see where it occurred. So again, from an upside standpoint, we're replacing an enzyme that's not working. That's usually a much cleaner readout. You're just replacing what's not there. It's an easier infusion, so it's not really brain surgery in the same way. So it's a little bit faster. It's open label, and with this mechanism, there is potential for disease modification in this very particular subset of Parkinson's patients. There's only GBA patients were included in this, and I'm very interested to see what the results look like from this readout, and something to keep an eye out for, especially if you know anyone with a GBA mutation. Okay, this is more near and dear to my heart, so I'll talk about this in a little bit more detail in a few minutes. But this is AskBio's program, so this is AAV-GDNF. We were looking in early and moderate Parkinson's disease. The study is also active, but no longer recruiting at this point. So like a lot of what we've heard, this is a phase 1b, so it's open label. Everyone knew what they were getting. And with GDNF being a growth factor, there was potential to regenerate the dopaminergic system and restore the dopaminergic tone to help improve motor symptoms. Again, neurosurgical delivery to the putamen, been a one-time procedure. So this study has read out the 18 months, just like we had seen earlier. And when we look at our moderate group, we see improvements in good on time by a little over 2 hours, less off time, less time with dyskinesias. And we also saw improvements on the UPDRS motor score. So again, this is how a neurologist rates how severe your Parkinson's symptoms are. And again, this is about a 20-point improvement that was seen here. So again, very significant change was seen in these patients. In summary, we found that this AAV-GDNF was safe, it was well tolerated, and we did see some pretty consistent clinical improvements, especially on the motor front. This was a small open label study, and I would say we were excited enough though with what we were seeing at this time point that we went ahead and started a phase 2 study. So this is active and recruiting right now, looking for patients most parts of the globe at this point. So this is a phase 2, and it's a controlled study. So just like some earlier ones I've mentioned, some patients will not receive active treatment so that we can monitor the placebo effect. Again, it's GDNF, so we're looking to regenerate the dopaminergic system and improve motor symptoms this way. The surgical delivery is identical to what was done in the prior study I just mentioned. And you can see some of the qualifications for the adults being entered into the study and where we're conducting the study. Some upsides, GDNF as a target has been looked at for about 30 years in Parkinson's disease. Using AAV, GDNF itself has been looked at for over a decade at this point. There is a lot of hope and potential for disease modification with this particular technique. And again, one-time procedure, no hardware left behind. So some big upsides to a gene therapy approach. Now, for some watch outs, the procedure is long. It takes a long time. We still need to, show long-term durability and these sorts of things. And we need to see how this compares with the placebo group, which hasn't been done yet. But again, very promising, and I'd like to dive into this study a little bit more in the next few slides. So AV2 GDNF has been looked at for, again, over a decade now for Parkinson's disease. And so I think it bears giving a little bit of background here and why we're so excited as Bio with our current Parkinson's program. So what's the link between GDNF and Parkinson's? So back in the 1990s, GDNF was discovered, and at that time, they found that you have to have GDNF in order for your dopamine cells to even develop in utero. So when you're an embryo, you have to have GDNF for dopamine cells to even begin to form. We also know that as time wears on, as we get older, our brains make less GDNF over time. And it's been thought for a while that losing more GDNF in some cases puts you at even greater risk for developing Parkinson's, and its loss is considered part of a contributing factor in the progression of Parkinson's disease as well. The hope with GDNF gene therapy is that this can actually, the cells that are still remaining, that are still healthy and still functioning well, if they can pick up GDNF gene therapy, they can then make a lot of GDNF. And I'll show you a video here in a minute, but GDNF is then able to help all the surrounding cells. And the idea is that, and we've seen this in a lot of preclinical work in animal models, with more GDNF around in the system, it's able to restore those sick and dying cells, or the cells that are kind of being hurt, haven't died yet, but they have a chance to come back. GDNF seems to do a really great job of bringing back those cells. And I'll show you some videos here in a minute. But before we get there, the one question I get asked a lot, it's like, well, how do you do gene therapy and how do you do it in the brain specifically? And so how does this neurosurgical technique look? So I put this picture here because right now we're doing everything inside an MRI machine, so we can be very, very precise. So we have these MRI monitored infusions, and that's how we deliver in a very controlled way our gene therapy. And so what you'll see here is we have one hollow tube that comes from the back of the head and is inserted very slowly in this direction in order to fill up this structure, the putamen, with our gene therapy. And it's on each side. So this happens on both sides of the head. This is administered in the MRI, and it's monitored all the time. So we're constantly catching new MRIs to see where we are and where our gene therapy is going. And this is done fully asleep. This technique has been used for over 15 years. So this isn't just for Parkinson's patients. There's a lot of other brain diseases where this has been used. So things like brain tumors, but even in some conditions in children, this technique has been used for many years. Get asked this quite a bit. So the surgery day is one day, just one day. There is a whole lot of work that happens before then and a whole lot that happens after that, but the day itself is pretty quick. Patients or participants in this particular study, they're usually in the hospital 1 to 2 days. It's not very long. Typically, if you're not taking anything out and you're not leaving anywhere hardware behind, that usually is a fairly quick recovery in that case. There are a lot of visits before, a lot of visits after. And I'll show you the schedule here in a little bit, and then a total of 5 years of follow-up that come. Now, what does this look like? So this is the current technique that we're using for the study. This is, let's see if this video will play. Ooh. Oh, come on. Hmm. Oh, well, that's too bad. Okay, there we go. So I'm going to show you, kind of orient you. So first, these are the eyeballs. So you can see at the top here, ears would be over here. And what we're seeing happen right now is there's this black line coming from the back of the head. This is that hollow tube or a cannula. And there's one here on this side, and there's one on this side. We just can't quite see it because it's out of the plane of view right now in the MRI. And what you can see is there's this kind of comma-shaped structure. It's kind of a darker gray here. That's the putamen, and that's the area that we're targeting. And so what you see is this little ball of white form and then another little white ball. This is actually the gene therapy itself that we can visualize in real time in the MRI machine. And so what you see happening is there's one little infusion that starts at the back of the putamen, and then we advance the cannula and infuse and advance and infuse and advance and infuse. So this is what we call the infuse as you go technique. And this has been really important in changing the whole procedure itself. It's really critical to get the full spread throughout the entire putamen, and this allows us to do it with just one pass of the cannula. It's shortened the infusion time and has allowed us to get over 60% coverage of the putamen, which is the highest we've seen it reported to date. So again, decades of experience have gone into this technique in particular, but I hope you can also appreciate how controlled and how precise this is as well. Now, once we get the gene therapy in there, this is where we get the AAV is really good at getting the gene in. The envelope goes away. All we're left with then is just the gene. It gets read by the cell, and the cell says, okay, we're making a lot of GDNF. The nice part about GDNF is that it's made and then secreted. And so the benefit is not just inside that cell, but all the cells surrounding it get to benefit from GDNF as well. So it's a big plus for this growth factor. And so this is what AAV1005 or AAV GNF looks like. So again, we have our shell. So this is our capsids, the AAV. We have a promoter that it promotes the production of our gene, and then our actual transgene itself, which is GDNF. And this was the phase 1b study that I showed you the results a little bit ago. So what we did is we split the groups into mild or moderate disease, and we based this off of how many years from diagnosis and how severe the UPDRS scores were. So how severe were the Parkinson symptoms? And you can see the differences here. As I mentioned, there's a whole bunch of neurology evaluations. So we look at UPDRS and a whole bunch of other scales and a lot of imaging. So we were looking at DAT scans, FDG, MRI. We had a whole bunch up front. So this is a situation where you measure 1,000 times because you can only cut once. This is a one-time procedure. So we're very careful up front. But then after this, we have participants followed up very closely in the first year, and then things sort of spread out after that. Right now, we currently have pretty much everybody in long-term follow-up at this point. Primary objective of this study was safety and tolerability. But we were very curious to know if what the effects were on the motor symptoms and actually non-motor symptoms of PD as well. And very curious to see what the changes in like DAT scan, for example, looked like. So just real quick on the safety. Right now, what we reported out at 18 months is that the AB1005 was well tolerated in both our groups. We did find that there were safety events that happened during the perioperative period. So this would be within 1 month of having the surgery, and then pretty much everything else that had happened. So sure enough, within the perioperative period, it's things like headache, for example, was very common. We drilled holes in people's brains or in their heads. Not surprising to get a headache here and there. But we also had a lot of issues with like joint pain from just being positioned, like shoulders and hips and things like this, that went away, shortly after treatment. And then post-operatively, it's things that you would expect, like dyskinesias picked up a little bit and required reductions in medicine, levodopa, but also things like COVID. We were conducting this in 2020, and so not surprising this happened there. I do want to point out that we had some changes in the MRIs. Hopefully you can see this teeny tiny little dark spot here. We thought this was likely to be surgically related, but small, asymptomatic, and really of no consequence over the past several years that we've been following this particular person. Now, from a data standpoint, this is where we got really excited. So I'm going to walk through this kind of slowly. So we're going to start here in this quadrant. So this is looking at that moderate group. So these are participants that were about 8 years on average from their diagnosis. Everybody's on, levodopa and, having, some motor fluctuations and issues with dyskinesias, things like this. They've been at it for a while. And what we can see is this group came in with about a little over 40 points of their UPDRS, which is pretty middling, and you feel that. But what we saw by 18 months is that this was down by about 20 points across the entire group. So that's about half of their severity of their motor symptoms was cut in half basically by 18 months. And what I think is really fascinating, and you don't see this in every type of study, but this was part three in their off score. So this is what they were doing, how they were performing, how severe their symptoms were without medication on board. So they've had no meds for over 12 hours, and this is how they were looking without medication. Now, what's interesting is you bring levodopa back and you have patients take their medicine. We still saw improvement. So they were responding even better to the levodopa as well, which I think is really unique and speaks to the mechanism of action of GDNF. Now, this part two, this is looking at how your motor symptoms are impacting your ability to do your daily activities at home. So this is not a very big scale, doesn't usually change very much, but we actually did see a small improvement here in this part two. And again, if you are improving the motor scores, you should also be improving how they're impacting your day-to-day activities. And we did see that trend here as well. Now, I'm going to skip down to the SMILE group. So these patients on average were about 2 years from diagnosis. So kind of give you some perspective. And again, looking at their part three off scores and how severe their Parkinson's symptoms were, it actually was pretty stable. We had this uptick at the end was from a patient that was found to have a TH mutation. So there's a mutation in the enzyme that's needed to make dopamine, for example. And so for her, she actually needed, her scores went up a little bit, but it was just her. And sure enough, when you look kind of across the board, these lines are very flat. And I'll tell you, don't have the data here today, but we have data going out to 36 months where, again, this mild group is really flat. Now, that may look really boring, but for a neurologist like myself, I think that's really exciting to see no change. I think a lot of patients would feel great if we could keep them where they were at baseline and not get any worse. I think that would be really remarkable. And so really excited to see what this data looks like, at 5 years, after, because we really haven't seen anything look quite like that in a mild group before. So it's one thing to have UPDRS. These are rated mostly by a neurologist. And so we also did diaries as well. So these are reported by the participant. Are you feeling off? Are you feeling on? Do you have too much dyskinesias? And we were able to mark this, and every 30 minutes, patients have to mark what they're experiencing. And so, as I mentioned earlier, we saw improvements in the good on time. So this means this is, your meds are on, they're working well, you feel good. And if there's dyskinesia, it's mild and it's not bothering you. But I think what's really cool is to see that there's reductions in the troublesome dyskinesia as well. So this is troublesome dyskinesia is when you have dyskinesia that bothers you is really what it comes down to. And to see this kind of melting away, I thought was also really interesting, in addition to getting less off time. Now, in a lot of other Parkinson's studies, there are a lot of medications, for example, that were used to bump up dopamine levels. And when that happens, there's always been a trade-off. Like you get the on, but you may get on with dyskinesia. And we actually really are seeing a much more normal physiologic type of change with GDNF, meaning that we see less off time, less troublesome dyskinesia, and good on time. And again, suggesting that we're really impacting the overall dopaminergic system in the brain. Now, we were very excited by this study and the results that we were seeing even by 18 months. And this led us to start this phase 2 study. So this is called Regenerate PD. This is currently active enrolling here in the United States. And as you can see as well, we got fast track designation and RMAT and a whole bunch of other acknowledgments from other agencies about this study. This is very similar to what we had seen before with regards to how the study was designed. Does that? So we're randomizing patients. So this is a blinded study. So for every two patients receiving active drug, one will receive a control surgery. We have patients then put into the active group or the control surgery group. Again, 1 day of surgery. And then, just like we saw before, there's a lot of very close follow-up for at least the first year. And then, after patients, we need everybody in this study to finish their 18-month time point, and then we can unblind the study. For participants who were in that received the control surgery, they'll be put into an open label study and offered the opportunity to receive active drug. For everybody else, there's a long-term follow-ups. We get to follow patients out even longer to make sure we can see the safety and the durability of gene therapy. Here's listed some of the primary inclusion criteria for this particular study. I have some information at the later if you have more questions about this, but just in just looking for 45 to 75 years old, about 4 to 10 years from diagnosis. Need to make sure it's Parkinson's disease and not other Parkinsonisms like MSA, for example. And we need patients that have been on a stable regimen for a couple of weeks. There's actually way more things to exclude people from this study, and primarily because we need to make sure people are safe and eligible to undergo a neurosurgical study. So again, it's a lot of things that if there's a lot of strokes or other what we call vascular changes in the brain, this is probably not a good candidate for brain surgery. Again, patients with significant cognitive impairment or frank dementia, this is probably not the right type of procedure for you. And this is very similar with the psychosis and other impulse control disorders. We are actually, potentially increasing dopamine in the system, and this potentially could worsen some of those features. There's also a lot of contraindications with MRI. Some patients have a lot of issues with claustrophobia, for example. There's a lot of MRIs in this study. It's probably not the right one for you. And then again, patients that have had DBS already, if the DBS is there, even if it's MRI compatible, because how much MRI we do, it will actually interfere with our ability to see. So we really are looking for patients that haven't had other experimental brain surgery or DBS at this point. Again, we're looking for at 18 months, and we're looking at how well patients are reporting they're doing with their diaries from the study. Mentioned this is where we're currently have all of our sites. So you can see we've tried very hard to spread around the United States. I'm sure many of you are familiar with some of these sites already. And you can see where we are currently in Europe. So UK, Sweden, Germany, and Poland are where we have most of our sites right now and covering a good chunk of Europe as well. There's a QR code if you have any questions. This is a great resource. This takes you to one of our landing pages, and then I'll have a few other resources here at the back end. And again, I just want to wrap up. Like, there is a reason why we need things like advanced therapies for Parkinson's. We haven't had much, innovation from levodopa since the 1960s. And it's very clear that we need a different way of trying to get at Parkinson's disease. And that's really what interested me in gene therapy and got me out into this area. And again, this is really, if we want disease modification, if you really want to change the disease course, we probably need something like an advanced therapy to help get us there. And that's why I love cell and gene therapy so much. I think there's real potential to regenerate the system in a whole variety of ways. Hopefully you saw a whole host of different modalities that are being looked at and explored right now in ways that were never even thought possible, even 10, 20 years ago. And I think there really is real hope to modify the course of Parkinson's. We've already seen this happen in Alzheimer's disease. Like, they've already made it there. We can do it here in Parkinson's, and we're darn close. And so I think the other big reason there's a lot of hope for cell and gene therapies is this ability to potentially just reduce the overall burden of Parkinson's by slowing things down. This helps not just the Parkinson's patient, this helps the entire family and caregivers as well. So if you want to learn more, again, I will say it again, just be careful where you're getting your information. Talk to your neurologist, talk to your neurologist, talk to your neurologist, talk to any doctor you want to, but you should talk to other physicians. There are some great websites out there. The Michael J. Fox Foundation has a really good trial finder that can really get you very specific to things you might want to participate in. I always encourage clinicaltrials.gov. It's a little bit harder to use, but if it's a legitimate clinical trial, it must be on clinicaltrials.gov. And so it's a great way to just check your references. But there's other companies and universities are sponsoring studies. So if you have, location, an institution near you, you could always go onto their websites. They often have trial finders there as well. But again, I can encourage you, please talk to doctors that you are familiar with and know you and your situation. That's really kind of critical. Um, Big thanks and big shout out to all the participants we've had in our study. This is, not many patients are, willing to step up. It's like 1 out of 10 will actually participate in clinical research. So this is my little play to say, hey, please think about joining the fight. Consider participating in a study. It's a big ask, I know, but we can't learn anything and we can't drive research any further if we don't have anyone participating in these studies. So thank you to everyone who's done it before and anyone who's thinking about potentially joining a study. And with that, I say thank you very much for all the time you've given me, and I'm looking forward to the questions.
All right, fantastic. Thank you so much. Very interesting information, very exciting technology. All right, some people have put questions into the Q&A feature, so I'm going to give you some more time to do that. I will say that we're screening out questions that don't apply to today's topic. And certainly you're welcome to follow up with us if you have questions that you need to get answered. I don't have any way of acknowledging the people that are trying to raise their hands. So just so that you know, I just don't have any way to communicate with you. I'm not intentionally ignoring you, but if you have questions, you will need to put them in the Q&A feature at the bottom of your screen. So, all right, Dr. Van Laar, we have a lot of really great questions. And first, let me also say this. This program has been recorded. So there have been a number of people who are asking, can we get transcripts? Can we get slides? But this will be available on websites, on IPN partner websites, once we get it kind of edited and ready to go. So you'll be able to find it probably however you found today's information is where you'll go to find that archived video. But you can always reach out through the Zoom registration and we can get it to you if you can't find it. So, all right. What about, okay, so I'm just going to kind of go through these questions. I thought I was going to be more organized about how I could ask them, but they're coming in kind of all over the place.
Yeah.
And a couple of the questions were related to people who have either dementia or people who have MSA or PSP, and whether or not one, there's any benefit to MSA or PSP, disease modification and symptom improvement, whether or not those folks would be appropriate and eligible to participate in the study.
Funny thing, we actually have a study that's active right now in MSA. It turns out that MSA is actually a GDNF deficient state, so that there are actually even lower levels of GDNF that we think is contributing to the degeneration in MSA. And so they behave differently, they move differently, and they can't really be scored on a Parkinson scale in the same way. And so we really have separated them out. But there is definitely very clear evidence for GDNF and MSA. Now, PD and MSA are very similar because they're both alpha-synucleinopathies, so the same protein is getting messed up in both of those diseases. And I think that's part of why GDNF has been looked at, and it's been somewhat successful in animal models. Now, in PSP, this is more of a tauopathy. It behaves very differently, and it advances very differently as well. In which case, it would be very confounding for us to try to understand what's happening in a Parkinson's patient if we included these other participants in the study. If GDNF works for PSP, we don't really have much data on it yet. But I don't know of many groups that have been looking at it. But MSA for sure benefits from GDNF.
Okay, and people with dementia, are they eligible to participate in this?
No. So
Trial?
we allow for, mild cognitive changes, but frank dementia and even DBS is the same way. Introducing a neurosurgical procedure on top of that can usually really throw patients off in not a great way. And so it's just usually not considered worth the risk.
Okay. And I apologize. MSA, for those people who are tuning in and don't know, is multiple systems atrophy. PSP is progressive supranuclear palsy. And so I should have used those words instead of just the acronym. So I apologize for that. All right. You talked about the cutoff age of being participating is 70 to 75. Why is that? Why does that seem to be the cutoff age, especially this particular person says he's interested and he's 78?
Yeah, I've talked to a lot of 78-year-olds in this very situation, and I know a lot of very, very healthy 78-year-olds. By and large, we have to put a line somewhere. It's a clinical study. This isn't an approved drug, and so we have to be very, clean with the rules, basically. And typically, from a neurosurgical standpoint, doing surgery over 75 becomes more challenging. I've seen it done, all these other situations where it's happened, like DBS, for example. But usually that's an approved product and it's, easier to make those benefit risk sort of decisions. We don't have that understanding right now with cell and gene therapy. So we're trying to figure that out. And as you saw, most of these are neurosurgically delivered. And so a lot of it's because of the surgical risk and the anesthesia that comes with it, that sort of thing.
All right. And you mentioned that people who've had DBS are not eligible to participate in the gene study. But do you see a potential for people who've had DBS, if this goes to market or even future studies, for people who've had DBS to be included?
It becomes a real challenge again. Like we actually can't see, right now we're doing this in the MRI machine and we actually can't see where we're going if the electrodes are in place. So that makes it, we just can't see. We're working on trying to scale up and do the technique a little bit differently. So maybe we don't need MRI, but this is not where we're at right now. And so I'd say it's still not a good idea.
Okay. All right. So in the Ask Bio procedure, can you describe how the insertion affects the area of the brain that it passes through?
Yeah, yeah. So, like I said, it's a measure 1,000 times and cut once type of situation. We have several neurosurgeons who look at the path. There's a lot of software that's used to help predict the trajectory of the cannula. That's again, so it's not done, just by any one person's opinion. So we are always looking for parts of the brain that are not eloquent. So we're not going into primary cortices, so meaning we're not going right into, in this example, to where your visual cortex is. We know where to avoid, or that are really special areas. Now, that being said, when we insert the cannula, you do it gently, right? We're not like shoving it in there. And when that happens, the brain is, like jello. It's very squishy. And so the cells, if you do it gently, the cells just kind of help part and they move aside. And because of the way the cannula is designed as well, it helps to kind of push the tissue aside. Um, and the infusion itself does the same thing. So there is minor damage, let's say just from inserting the needle. The same thing happens with DBS though too. I mean that needle, the probe stays in there. Now we're following and seeing what this looks like in the MRIs of our participants that have gone through before. In some cases, you never see this. It doesn't show up, after a year. In some cases, you see a little bit of a line that stays there. But again, this is a new technique, and there's really no one else that's been looking at what happens to the brain after you've put an instrument in there, for years and years and years. And so we are doing that so we can understand, what this looks like in a healthy person.
Okay, thank you. All right, sorry, just
Okay.
my questions are moving as people are submitting.
Good.
Um,
Good.
all right, so if a person were interested in participating in a trial, how do they go about? You showed the map, and so those are the places that the trials are being conducted. But let's say that you're not in that area, or you're working with, an institution or a clinic or a physician who is not involved in the research trial. How would a patient go about, inquiring about this or getting more information or actually getting enrolled?
Ask first at askbio.com so that we have all that information here. And I'm sure Pooja can send you some materials and links and things too. We have a new web page, which I don't have up here, which I think is a good place to go. But this QR code should help participants find this too. And we'll be sure to get you some more information when we get done with this call to send out if you don't mind. And then I would also just talk to your family, talk to your neurologist, and see if they, know of anything because we sometimes we get reached out by them as well. But Ask First at askbio.com is probably the first place to go.
Okay, great. And one person submitted said that they are a patient of the Blue Rock phase 3 trial. So would the AAV2 GDNF be a complementary treatment to the Blue Rock therapy?
It's a great question. There is actually a lot of research that's been done looking at exactly this question. We're not there yet right now. We need to know how each one of these, drugs is doing on its own before we can even think about doing a combo. But it's not the only time I've heard that question, so it's coming from a good place.
Okay. And if the person is interested, they're kind of looking at both cell therapy and gene therapy. Is there any one particular kind of frame of thought or line of thinking that might help a person decide which might be more appropriate for them? Or
Yeah. It's a tough question. And I think this really comes down to the individual. They're so similar and yet there's some key, differences that I think really needs to come down to like talk to your doctor and then talk to your family. Does it make sense for you and where you're at with your Parkinson's? But I know this question is going to come up more and more, but frankly, it's just great to be able to have the options. We never had options like this before for Parkinson's patients. So I'm just glad we have more tools in the toolbox to pull from. But again, I think we're also still so early in both programs that, I think who is the best patient, we're still trying to figure that out too.
okay, and you mentioned a couple times about doing your homework. There are several questions about either the cell therapies that are being offered in places like Mexico or some of the companies that are offering gene therapies. And so realistically, like, can you talk about the efficacy of those or whether or not those are well vetted and and meaningful treatment options?
Yeah, this is why I actually have that slide in there whenever I talk about these things because there's a lot of medical tourism, and I personally had a patient once that got injured, and so part of the problem here is you don't know where the cells are coming from, you don't know how they're being processed or treated, and so I've heard horror stories about people have actually requested some of these cells, and when they tested them, like everything was dead, and so you're actually just injecting dead cells into someone, and that's very dangerous. And people have died from things like this because the rigor is just not there. And I've even heard like there's clinics and here in the United States that do things like this too, and they'll take your fat cells and just inject them back into you. And I'm like, this, how is this supposed to make sense? Brain is so precious, and like putting something into an IV and expecting a brain cell to make it all the way up to your brain and into the right spot and then incorporate and do its job and make more dopamine. I think that's asking way too much of any cell. And so again, mostly the IV delivery, it just doesn't make any sense. This is why everything I just showed you pretty much requires neurosurgical delivery because cells just don't migrate to the brain. You don't want them to do that. That's a very dangerous thing. And so, this is also why I always emphasize go to clinicaltrials.gov. Like if it's a legitimate study that's got the rigor of, the FDA watching over them, it has to be on clinicaltrials.gov.
And could a person's participation in those types of gene or cell therapy preclude them from participating in research studies?
Yes, it
Okay.
could. It could.
All right. A couple of things. You mentioned fast tracked. What does that mean?
Yeah. So there are some, especially with gene therapies, there's a lot of potential. We're seeing, very big effects, from these studies because, again, if you're doing brain surgery, you better have a, notable effect here. And with that, showing that you've got a really strong drug, let's say, that then, allows us to move a little bit faster in development. So we got fast track designation, we got RMAT, we also are looking at, other sort of mechanisms in other parts of the world to speed up our development. And so, meaning that, maybe we don't need to follow the same rigor that we would need for other types of Parkinson's, like pills that you would take, for example. That we get special, attention from the FDA, for example, if we need certain meetings where it makes it easier for us to talk to the FDA more directly. And again, just kind of speed up development overall. So it's a big deal, really for us. And I would say for patients as well, because this helps us get, these drugs, through a little bit faster than traditional development.
All right, thank you. And lots of questions about, how far away do we really think this treatment is from FDA approval? And then, following, if you had a successful stage 2 study, how long does it take at that point? Do you still have to do a stage 3 study? This person also wanted you to know it was a wonderful presentation with very clear and very clear science to understand. So thank you for that. But also wanting to, know about how this, how far away we really are from this being FDA approved if everything goes as we think it or hope that it will. And kind of how does that do you have to do the phase 3 if you've had a successful phase 2?
I mean, typically in Parkinson's, because it's such a large disease and it's slow, typically the agencies, these regulatory authorities, they want, long, slow studies and, that cover multiple stages and multiple programs over years. This is again part of the negotiating that we're doing right now with the agencies, with the blessing of the RMAT and Fast Track and the other and ILAP, for example. So again, we're in the midst of negotiation. This really is going to have to hold the signal that we saw in the phase 1B needs to really hold into the controlled study that we're doing now. I wish I had that crystal ball though to give the number of years until we're ready. But really, it's a little bit tough right now to call that out. And we really just need to finish this phase 2. I think it'll be really clear at that point.
All right. Have you found that so far in the gene therapy and maybe what you know about the cell therapy, are either of those benefiting non-motor symptoms?
Mm. So I'll speak to our program in gene therapy with GDNF. So we saw some hints. Again, the phase 1B study that we did, it was open label, but there were some hints. And I would also argue, from what we know scientifically, we actually know that GDNF gets back to the substantia nigra, back into the midbrain. And I think that's actually where a lot of the non-motor control happens. And I think that may be why we're seeing a little bit of benefit there, but we need a much bigger study. So I mentioned like that part 2 EPDRS with the activities of daily living benefit that we're hoping to see. That's actually one place we're looking, but there's a lot of other non-motor scales that we've built into the study to try and measure this more directly. It's really hard to move non-motor symptoms, though, as I think probably everyone here knows. So it's and because of that, like we really just need larger numbers in order to see if there really is a trend there or not.
Right. Well, that's probably accounts for why so many people have asked.
Yes, I hear
Because
you.
yeah, that's the part of life that kind of creates a lot of additional daily headaches and challenges.
Well,
So,
I'm going to bring up sleep specifically because I feel like sleep impacts everything, right? And so if we can see sleep get better, for example, I think that would make a lot of sense. It fits with, sort of mechanism of action on top of that and would have a, broad impact across many other symptoms. So I just don't know yet, but I'm hopeful.
okay. Well, one person just put in the Q&A that there has been a non-motor benefit, at least for them, that their sense of smell returned and their constipation improved.
Interesting.
So yeah, so there's a little bit of
Yeah.
anecdotal
Yeah.
hope there. All right. This person has asked, one of the basic challenges with Parkinson's is that by the time of the initial diagnosis, many of the patient's dopaminergic neurons have already died. Stem cell treatment offers hope to replace those dead cells. By contrast, does gene therapy offer any hope to revive those dead neurons?
So if they're dead, they're dead and you can't get them back. But as I was saying, there are a lot of cells that are sick but not dead. And that's where I think GDNF is really has a unique place. And so in general, we need cells that are still there. So this is why moderate or early disease may be better for something like gene therapy, where you still have some cells to actually have an impact on. So in fact, what we've seen, we see this in mice, we saw this in monkeys, and I would say we've actually seen this in humans in our very first phase 1 study. Wherever GDNF goes, we see increase in sprouting from those cells that are still there, meaning that the connections expand whenever GDNF is present. This is where you get more channels to make more dopamine. You can handle dopamine better, and this is where I think we're really seeing improvements in the off time, the UPDRS scores. We also saw improvements in how much levodopa was needed. There was actually a decrease in overall levodopa in that moderate group too. And all that kind of together tells me that that's what GDNF is actually having a major restoration effect on the system. But instead of putting cells in there that are from somewhere else, we're just trying to harness your own cells and make them work better and not die so easily, so that some of the ravages of Parkinson's can't hit it as quickly.
Right. Okay. Um, is this the first iteration of gene therapy? And if not, what's different this time?
Mm. There's been several other gene therapies that have come through in the past decade or so. Some things that are the same. So that shell, the capsid, it's the one we're using is AAV2, and it's the same one that's been used in the brain many, many times. The delivery technique is really what's different. So as you saw the video with the white that was growing, that's really unique to what we do. And that means we know exactly that the drug's getting in, we know exactly where it's going and where it's not going. So it's very controlled. And I would argue that's really what's made the biggest difference. Most other gene therapies that have been done in the past was like a black box. There was no MRI, there was no visualization. We didn't see, we hoped we got to the right target and we put the right amount in and it got to where it's supposed to go. And we just didn't actually know but the delivery piece has really been the biggest difference. I think the other one for gene therapy too is coverage. The whole point of gene therapy, it's not really the drug you're putting in, it's the protein that then gets made that is actually doing the work. And the more cells that pick up your gene, the more protein you're going to make. And so that coverage is really, really critical. And it took us a while to figure that out. But I think we've got the right drug, the right delivery, the right time, the right patient. I think we figured it out. And that's what we put into the phase 2.
Okay. All right. I think we are down to kind of the time that we have remaining for one last question. And this is related to why can't a patient be unblinded after they complete their 18 months? Why are they required to wait 18 months after the last patient is dosed? What would be the
Yeah.
reason for that?
So this is scientific integrity, really in a nutshell. So I personally have pushed the FDA on this before. And because it's Parkinson's, because it's a slower disease, and because the placebo effect is so strong in Parkinson's, these are some of the major reasons why. But also from just a scientific standpoint, it is best to keep everybody blinded. You unblind one person, it jeopardizes the integrity of the entire study. And at the end of the day, folks like the FDA, they really want to know as much data as you can possibly give them that's been blinded. It's not as good, really, once it's been unblinded. And so this is why we're trying so hard to bring awareness to the program, because we'd like to get this recruited quickly so we don't have people lingering, in this limbo period, let's say. But this is so important. The placebo effect is huge in Parkinson's, and then you add a neurosurgical technique on top of that, it can be even larger. I personally have worked with people who thought that they had received active drug for 2 years and did not. That's how strong this can be in PD. So again, we really want to know what our drug is doing and not what Parkinson's or the placebo effect underneath is doing. Again, we want to make sure we're making the best product for patients out there.
Right. Okay. All right. Well, Dr. Van Laar, thank you so much for your time today. Thank you to Ask Bio for sponsoring this program. As I mentioned earlier, this has been recorded. We will be doing some editing, and then we will get that out to folks so that you can access this and watch it and rewatch it and rewatch it until you've memorized the whole thing, and then you can go out and talk to all your friends and colleagues about the importance of research and moving science forward. So thank you all for joining us. Thank you, Dr. Van Laar, again, and to Ask Bio. And we will give you back the rest of your day. But thank you for joining, and thank you for all of your wonderful questions.
Thank you so much for the opportunity. The questions were amazing. Thank you so much. Appreciate
Yeah.
it.
All right. Goodbye.